Good afternoon, everyone. Let me share my screen. Okay, so today in Python, we are going to explore the very important library that is nothing but BioPython. Okay, so BioPython is what? BioPython is nothing but collection of Python tools, the tools which we want to analyze biological sequence. So this is nothing but the tools basically used for computational biology and bioinformatics. So it contains all functionalities for working with biological data in the sense like sequence data. You can consider DNA, RNA, amino acids, or protein sequence. Okay, so there are different types, different formats of the data, even expression data, gene expression, which is in numerical format. Okay, so what all things we can do with BioPython? So as if you are working with any environment other than this Colab or in Colab itself, we need to first install BioPython because that is not the repository which is pre-installed. Okay, in any environment we have to install this. So how to install? By using pip command. Pip install, as you already installed some libraries which are required for data manipulation like NumPy, Pandas, Matplotlib, right? So here you need to type command pip install and the name of package, that is BioPython. Execute this. So it will fetch all the functions, packages, dependency packages and all. It will show the collecting BioPython packages, and finally you can see the message successfully installed BioPython along with the installed packages. So this way you can also install. Now in BioPython, there are few we'll see few modules along with few classes, because in BioPython there are many modules like which are required for many kind of analysis like sequence analysis. If you want to fetch database, if you want to do a phylogenetic tree, or just want to work with the protein data. So depending on your requirement, you can use that particular class and that particular module. Okay, so simply if we are working with sequence, so what all things we can do? We can create a sequence. We can do this the process that is central dogma: transcription, translation, reverse complement. Even we can count individual base frequency for each in base, nucleotide base. Okay, even you can calculate the GC content. We are looking for if motif or subsequence, that also we can do. And also we can concatenate, like connection between two strings. Okay, so these all things we can do for sequence. Let's see what all modules are there. So for that, first important thing, we have to type bio, because from bio we are using that different modules. So bio dot, there are many modules like bio.sec is there, which is useful for handling sequence data. Okay, so with this sec module we can create our sequence, or we can create mutable sequence, which is sequence similar to sequence, but some modifications of the sequence after creation, we can do that. Then there is one module called as bio.sec record. So sec record is for, we can check all the records of that sequence. Records in the sense all metadata, all sequence information like the metadata, annotation, even features, any database references. So this is nothing but when you want to handle the data with the context, then we can use records. Also there's a called as align, for alignment. Either you can align two sequences, that's pairwise alignment, or multiple sequences, multiple sequence alignment. Then bio.pdb is also there. PDB is for passing PDB files, or there are two functions in this, like either PDB parser or MMCIF parser. So PDB parser is for parsing or information. So in PDB we are getting structural information of biomolecules, right? And which is very crucial for structural bioinformatics. So we can pass the PDB files. And there is a, if you are looking for database, then we have to do bio.entrace. So with entrace, we can fetch records from entrace database, and entrace like PubMed we can use, or even just want to search something, you can use eSearch. For alignment purpose, just call blast similarity search. Okay, we can do similarity from NCBI standalone blast or NCBI www, anything we can use here. Then for phylogenetic, just call phyllo. Even we can fetch swissprot data like this. Those who are interested in pathways, they can fetch KEGG. KEGG is for pathways analysis. You can analyze pathway, you can analyze function and annotation. Okay, there are many more. So this is all about different modules and different classes. Okay, so let's fetch one of this. We'll first create a sequence and then we'll proceed. So how to import, as we have already installed this biopython. Now from bio, so from this bio.sec, this is the first one which we discussed. From this bio.sec, what we this line is basically for sec class. Okay, so we need to import sec class. So sec class from bio.sec module. Okay, this is from biopython library. So what all things we can do here? We can create a sequence. So let's create a sequence. Simple sequence. We can create a DNA sequence or RNA sequence. So if you create a simple string sequence like we have created previously, a simple string like this, let's call this sec. Okay, and then check what exactly the this sec variable type. So this is str. Str means string data. But same sequence if you are creating by using sec function, like in new cell I'll create, so you can see the difference. Okay. Yes, any query? Just kindly mute. So you can see here I'm creating one new variable as a biosec. In that sec we are fetching existing sequence but with help of sec function. Or you can, if you don't want to use previous function, you can directly mention your sequence like this. Otherwise, we have already declared this sequence with name sec. This sec we can use inside this sec, small sec. Okay, this sec is function name. So now let's call this variable, and then if you want to check, you can call this is a sec with sequence. Even let's check the type of this variable. So type is what? Bio.sec.sec means this is a sequence data abstract base, and you can see this is the sequence which we can use for, even there are many functions. So to check functions, we can call directory of this bio.sec. So these many attributes are present. So what exactly attributes? Like you can do, these are functions, these are attributes. So you can see back transcribe is there, complement RN is also there, counting overlap, then remove prefix, reverse complement RNA. Now let's check the dir directories for simple sec which we have created. So there are, these are the attributes. So we are not getting any advanced attribute which can deal with the sequence like reverse or related to sequence. Only we are getting with the string related functions, not sequence related functions. Okay. So these all things will be appeared here. Clear the difference? Now let's do some let's first create sequence. So here I'm providing a name, DNA sequence, or let's make it short, DNA sec. This is sec, and here I'm creating sequence. You can also create your own sequence. Now time, this is DNA sequence. So we can transcribe this sequence, means we can convert this DNA into RNA. So for that we need original sequence, or we can create a variable. So in that variable we can store the RNA sequence. So let's fetch original sequence and then use function transcribe. Okay, execute and check. Okay, now you can see the output is a RNA sequence. Now we have RNA, so let's translate this RNA into protein. So for that we need input as a RNA sequence. So take RNA sequence dot, we can use function translate. Also we can store in the new variable, like this is now our protein sequence. Okay, let's check. This is randomly generated sequence. So let's see what proteins are we are getting. Okay, so this is the list of amino acid. Okay, now what else we can do? We can complement the sequence, like so let's create your cDNA and take sequence and use function complement. If you are working with RNA sequence, you can see the option RNA complement for RNA, reverse complement RNA. So you can try these all. Okay. So this way we can create a sequence, we can generate a transcribed sequence, translated sequence, complementary and reverse complementary. You can try for reverse complementary. Now let's see. Suppose we want to fetch information from M3's database. Okay. So like we should you should know the ID, or one thing you can fetch the sequence by providing the gene and their ID name, ID if you know. So that thing we can do here. So let's create first. You need to provide one email ID just for verification purpose. So this NCBI will give access to fetch the sequence. Okay, but for that we need to fetch first module. So from bio, first we need to import this class and trace, then we can work with this. So when we call this entries, we can fetch different functions. Like if you write it here entries and dot, you can see all these functions we can use. Like you can fetch the sequence, you can eg query, e-info, e-link, email. So to declare email we can use email. Here I am writing some demo mock name. So basically this email id provides the access to the NCBI database. That's why we need to mention this ID. Okay, now we can use this, or before that, let's create one gene ID. So for that gene, we can fetch the information. Okay. So whenever we are fetching, we need to read line by line. So let's create a handle to read that data and then we can read. So let's create a handle by calling entries as we want to fetch. So we need to call efetch. There are e-info and e-fetch. E-info to just get the information about the details like database. Here we are fetching, so just mention. You can see here how to fetch. First we have to mention the database. So let's take a gene database. Then ID is nothing but, as we already declared the ID. So what we can do, we can take this gene ID variable. And also you can see here the reference, how to declare, how to fetch. So in Python, you can use this pattern like from bio entries, import entries. You have to provide the name, email ID, handle. Also, you can provide ID directly, or you can create ID variable and fetch that ID. Retrieval type, retrieval mode, and then we can read by using handle read line. Okay, so let's provide retrieval mode. Not necessary to use every parameter. Those which are required, that only we can fetch. So here we have use XML. After that, time to read the data. So here I'm creating one variable, or let's create gene data. Call this handle because in handle every information is stored for this data, and then call function read. So we can read the content and then execute. So for execution, call this variable, or you can call print function. Along with print also you can execute. Okay, so this is all about information. So from this, so this is XML file. That's why we can see like this. You can use, there are many file formats are given like text and HTML. So you can select any one of this. Now how to work e-info. Like same way, we suppose we want to fetch, this is for gene we did. Now let's take example for, let me copy this. Okay, so first two lines are same because we are importing from M3s for this email ID. But now as we want to fetch information, so we have to use e-info rather than e-fetch. Okay, so also we can create a variable like rather than handle, let me generate a new variable like database information. That information we are fetching from e-info function. So what information we want? Let's write about PubMed. This database is already done, right? So we can take that. And then same way we have to read this information. So any variable you can create gene data, or let's use the another name. So this is a database. Just need to read that data. So call entries dot read function, and we are reading this database information. That's it. Call this DB to check the output. Okay, so this is a huge data set. All of you get it? So you can read here database information. Database name is PubMed. Menu name is PubMed. Description is what? PubMed contains all bibliographic information, like all literature and every detail about the articles and things. So this is all description. Just scroll down, you'll get a detailed information. Okay, so this way you can find the information of any database. Done. Okay, now suppose any sequence you want to read, like FASTA sequence. You can consider any FASTA sequence. If you don't have right now sequence, so let's open this directory. This is a current working directory. You can create a new file. Okay, and here you can create one demo.fasta sequence, and you can write some lines here itself. So how to write FASTA sequence? This is greater than sign, ID, then second or description, and then you can write sequence. Okay, mention that sequence 1 on second, or if you want to specify a description, that also you can specify, and then write 2 3 lines for sequence. Just let me know after completing. Or let me fetch any sequence. Suppose this one sequence I have fetched. So first line is what? Greater than sign, session ID, the description, and a sequence. Okay. So for this sequence, let's pass the sequence. Means read the sequence in detail. Means not necessary to read whole sequence on the terminal or on the page on this environment. Better you can fetch one by one. Like if you're just looking for ID, if you're looking for a description. Okay. So that also we can do. So for that, we need one more class, that is sequence input output, as we are going to fetch sequence. So import sec input output. Okay. Now which file you want to pass, that file you have to fetch first. Suppose we are creating here one variable as a FASTA file, and let's fetch that file. So what can I do? Let's copy directly path like this. So this is our file. Now this file we want to parse. So parsing is a function. So let's create a variable records, or any name you can give, call function, and then here we have to mention which file we want to pass. So this is a file, and this file is in the form of FASTA file format, like this. If it is FASTA, if it is text, depending on, you can write. Right now this FASTA file content, the content is FASTA format data, but it is a text file. So you can write it anyone. After that, let's do one thing. Let me copy this file, and we can create 2 3 sequences here. Like this is one sequence, 2 3 4. Okay, so these are different sequences, or just change the name here itself. Save. Save and execute. Okay. Now this file we are going to fetch. So this file is what? Demo.fasta. So copy the path and then provide path here. Okay. Now our file is what we are fetching, that is demo file. Now after passing, what we need to do, read the content of the file. Okay, so for that, let's read one by one. So we can use loop, use it a variable as iterator inside this records. Okay, so this is for now. After iteration, what we want to do, like we want to fetch information. Okay, or simply we can print record. Let me call record first, then we'll call ID, description, and sequence. Okay, so now here you can see the sequences we have changed, like 1, 2, 3, 4. So the 4 sequences we are getting like ID, name, and description. Can you see? Let me open this file here. Okay. So in this file, what exactly the ID? ID starts with greater than sign. The sequence, this file starts with greater than sign. So this is the first ID. The description is what? This sample, species, and then sequence. If there is any feature, so this will also display, but there is no feature, so we are getting 0, 0, and the actual sequence. Clear? So this way we can fetch the information from any sequence file, pasta file. Okay. So after this, this is with help of sec.io. Okay. Now suppose you want to check or you want to align the two sequences. If there are two sequences, means we can perform pairwise alignment. So for that, what we need from bio. Let's import new class, that is pairwise. Here you can see pathways, APSCHE, pairwise 2. But we don't have sequence, so let's create sequence here itself by using sec module. This is my first sequence. Let's copy the sequence. You can create another sequence or rename this as a sequence 2. If you want to change some basis, you can change it. Like, let me cut this and paste it at the bottom. So these are two different sequences. Execute. Now these two sequences we want to compare by using pairwise alignment. Okay, so there is a standard syntax. Like first we have to call a method like global, local. Call this pairwise. As we want to align, we can call align function. Now specify method global, local. According to that we can call here. So these are the methods, and then provide the input, the first one and second one. Okay, one more parameter you can add here, like you can see here global XX having sequence A and B, and you can read here in detail. So A and B are what? Strings, which shows the alignment between the sequences. So for that, if you want only one alignment, so you can call this parameter one alignment only. Let's keep this parameter true, and then read all the alignment. So for reading, we have to iterate because we are not sure like how many bases are there, how many lines are there. So better we can create a iterator or a variable, and that variable we are iterating inside this. Let's create here. This is nothing but our alignment. So I'm renaming this as a alignments. Okay, so this alignments variable content our alignment. So we are iterating inside this to check the outcome. Complete the for header and then execute message. So first call this variable alignment and first sequence. That is nothing but sec a. It's written in syntax, right? Same way we have to call another function, another sequence. Okay. So this is the alignment. Clear? Or simply we can call alignment, like rather than writing this. If you're calling simple alignment, so we'll get in this fashion. Sequence A, sequence B, score, start and end. Now you can try for local. Let's copy this sequence. Rather than this global, I'm changing the algorithm. Okay. So are you aware about this local alignment and global alignment difference? If you want to check score, so that also you can check. So need to call score like. So the score is 13. Clear? You can try for any two sequences in the same fashion. First create two sequences with sec function. Any query till here? No? Okay, let me show one more uh, function that is nothing but align. Like here we have imported from bio import pairwise 2. We can call from bio import align. Also from bio dot sec, because we need sequence. So from bio dot sec, again we can import sec. Okay. So first let's call aligner, as here we have called pairwise 2. So we can call pairwise aligner. Let me show here. Okay. So when you want to call this align dot pairwise aligner, and also need to define method, like by which method you want to align. Then you can create two sequences by using sec function. Let me create here itself. Okay, so these two sequences we are going to align. So how to align? We need to call alignment. So for that, let's create, suppose this is an alignments variable where we are going to check alignment. So here I'm calling alignment aligner. Let's call aligner dot function align. For that, we have to provide these two inputs, sec1 comma sec2. Okay, to check the alignments, let's iterate. So for iteration, call this variable inside variable alignments. Okay, and then execute. Even you can call score. So if you want to display score in a proper pattern, you can use string formatting like sf, and then this is a label, and then fetch that score. So to fetch the score, always use curly brackets and call from alignment score. So score is what? 13. If you want to check alignment, simply call alignment. Clear? Yes? Any query till here? Okay, let me share a tendency. You can just copy this, execute. If you have any query, you can ask. You can text in chat box. Attendance sheet is shared in chat box. Kindly mark your attendance.